Thank you. And this is the last talk of this course, and I would like to thank the organizers for inviting me again. And I hope that so many speakers are informed you about the hybridization, gave examples, but now I will try to be more faster than I expect. So today I will go through the hybridization techniques and their practical applications. I will start with the definitions and explanation of the technique and also interpretation and applications of the diagnostic and research tools. You know all these terminologies, so it's very easy for me. I will skip all these things, but first of all, hybridization, you know that this is a process of establishing non-covalent hydrogen bands between two complementary DNA or RNA nucleic acids chains. So during the hybridization, hybridization can be between DNA and DNA, DNA or RNA and RNA. You know what denaturation is, so it is the separation of these double helix complex, and annealing is the sort of renaturation. So these are the important issues for hybridization. And probe is the key component of the hybridization. This is a fragment of labeled DNA or RNA chain, and it has got different length, and the length of the probes are usually longer than the length of the primers, which are used in PCR. So you need to have larger or longer bases or molecules, and which I will go through the importance of these lengths in further in my talk. That this is very important. You have to know what you have to know all about your probe. So the hybridization, in the hybridization, the main principle is the visualization of this reaction. So you have to visualize this hybridization reaction. Different hybridization techniques, many of them are also pointed out in the previous speeches. Blotting techniques, array technologies inside hybridization, which is going to be my main topic, and comparative genomic hybridization. These are all techniques which are depending on the hybridization. Blotting technique, as it is explained you before by Gaye, you fix your target sequence on a membrane, and then you apply your probe to this membrane, which is suitable for visualization. The array technologies are the reverse of this. So what is it? In the array technologies, your probes are fixed on the platforms or fixed on any platform. It could either be a glass platform or another platform. Then you apply your target DNA or RNA to these platforms, and you got this hybridization results. In the array technologies, you have to scan the information, the hybridization, by sophisticated scanners, and the interpretation is also done by these scanners and by sophisticated software. So you have to, you need to know a lot of statistics, and in order to interpret the results. And you have to know, and you have to be very aware of which tissue or which material you were using in order to interpret the results. Otherwise, it's going to be a mess. Comparative genomic hybridization, or array CGH, also Dr. Hunt has talked about it a little bit. This is something that you can examine all the chromosomal abnormalities in a cell or in a in the tissue. This technique is depending on comparing the normal tissue, normal cell chromosomes and the pathological or tumor cell chromosomes. So by comparing these chromosomes, you can tell that how many chromosomal abnormalities are present in the tumor tissue. This technique can be used and very powerful technique, but there are some limitations which, for example, this balanced chromosomal abnormalities such as reciprocal translocation or polyploidy cannot be detected by this technique. But if the abnormalities are unbalanced, then you can easily show and you can detect these sort of abnormalities by CGH or array CGH. Now we came to the in situ hybridization. So many of you, I assume, are pathologists. So I assume many of you are aware of or are using in situ hybridization. How many of you are using in situ hybridization in your laboratories? Good. So it's easy for me to explain it. I just briefly go through the in situ hybridization. So the principle is the annealing of the labeled probe, and your material is the cells or the tumor cells on the tumor tissues which are our material as the pathologists in our daily routine practice. In situ hybridization has started as an important tool by the cytogenetic analysis and by examining the human chromosomes in metaphase during the mitosis. And spectral karyotyping is this multiple hybridization of the chromosomes, all the chromosomes, and this is a sort of immunocytochemistry for G binding. This is G binding, this is H&E of the cytogeneticist, and spectral karyotyping is the immunocytochemistry of the geneticist. So, in situ hybridization for pathology, we have different material than the geneticist. We have cells in interface. That means that they are cells which are sleeping, and they're not in. Usually, most of the cells are not in mitosis. So. So interface fish is very important tool and became a very important tool for pathologic diagnosis. So what could be seen by using in situ hybridization? We can see gene copy number increases, amplifications, deletions, losses of the chromosomal parts or translocations. We can show we can see infectious agents in the cells. We can even see protein expression indirectly by showing mRNA using in situ hybridization. In transplanted patients, chimerism can be examined by in situ hybridization, and now microRNAs are becoming important, and in situ hybridization is also useful for detection of the microRNAs. So probes are important for in situ hybridization, and there are several types of probes. They could be double-stranded, single-stranded. They could be DNA probes or RNA probes, oligonucleotide probes, and peptide nucleic acid probes, which are more stable and handy for different hybridizations. There are several labels for probes, so the labels are important in order to decide which instruments or how you examine your hybridization, and also it is important for the sensitivity of the results. So there are biotin, avidin, fluorophore conjugated labels. There are also enzyme conjugated labels, hapten conjugated labels. These are all working like immunohistochemistry. The importance of the probes which are fluorophore, I will explain later, and the chromogenic probes are important because the signal that you have by in situ hybridization is usually a low signal, so you have to extend the signal. It is very important. These are all the non-radioactive probes, but in the past, radioactive labeled probes were also used for hybridization techniques. Probes can be synthesized by different ways. SNP translation, random priming, PCR are the methods that is useful for probe synthesis. As I said, 100 or 1,000 base pair length probes can be used. Longer probes maybe has got some benefit for increase the signal, but they have a disadvantage of the background staining. Shorter probes can be difficult to detect if the signals are in low levels and the target is in low levels, then the labeling can be very low. So we have to pay attention about what we would like to see and which products we have to use. Fluorescent in situ hybridization is done by the fluorescent labeled probes. There are several different fluorescent labels. Some of them are more stable, some of them can decay easily. So for fluorescence labeling, you have to examine your hybridization, and you have to fix your hybridized image, or you have to be very quick to detect these hybridization results. But chromogenic in situ hybridization, if you optimize it for different purposes, then it could be more easy for the pathologist. It doesn't need any other instrument. Your light microscope can be enough for detection and evaluation. So this technique is much less sensitive than fish, but it's very useful, especially if you would like to see some viral infections in the cells. Then it is very useful. Or amplification signals can easily be detected and more useful. Even if you're using suitable probes, and if you extend your probe signals, you can even use it for translocation detection. So probe signal as amplification is important. There are several different probes or systems that you could amplify your signal, and these amplifications are making life easy for the pathologist and increase the sensitivity of the hybridization. So if you would like to have a good visualization, then your target must be accessible by the probe, and the target must retain in situ and not degraded by the endonuclease enzymes. So DNA is more suitable than RNA. So when you would like to show the RNA, then you have to be very patient. It is very labeled. It is very. It can decay easily, so you have to be very patient. For what we use for in situ hybridization, culture cells can be used, aspiration smears can be used, fresh frozen sections can be used, and paraffin sections can also be used. I will try to focus on paraffin tissue tissues, because we usually use paraffin tissues, and this is much more tricky than the other tissues. So for inside hybridization, you don't need a very large space, so only a small bench can be enough for you to do all your hybridization. There are some fully automated instruments that now they are more handy and you can use them. As it is explained in previous talks, the tissue fixation is very important, as it is in the, for the other techniques. So your tissues should be very well preserved in order to get a good result. So we have to deparaffinize, we have to increase the probe incubation, probe penetration, and denaturation, stabilization of the denatured DNA in order to start our inside hybridization. So this deparaffinization is the step that is done in every start of the histology techniques. So the tissues, the cells are placed on the tissues within a matrix, and also there are so many proteins, and also the fixatives, they are all complex in the tissue sections. So you have to remove all these things during your pre-treatment. So removing lipids and some of the other matters is important. Detergents and HCl is used for this. And also we need to remove matrix proteins, and also we need to make some pores on the membranes which allow the probes to penetrate to the nucleus, our target DNA. Detergents and enzymes are used for this. And in order to start for hybridization, you have to denature and stabilize your DNA as denatured within the cell. So you have to, heat is used for that, and also formaldehyde containing buffers are used for stabilization. These probes can be single-stranded or double-stranded. If you have if you're using a single-stranded probe, then it is easy. Just drop it on the cell or the tissue and seal it, leave it for incubation. You can leave it for incubation. But if you have a double-stranded probe, then you need to denature your probe first. So this denaturation is done similarly as it is done as the target DNA. So you need heat and also formaldehyde containing buffers. You can do this denaturation either separately on the tube before application to the cell or the tissue, or you can also do it in situ on the tissue at the first step. If you're doing in situ on the tissue, then you need to heat your tissue with the probe first. So it's like a PCR reaction for annealing, for denaturation, and then the incubation starts. So when you incubate your probe, you need to increase the temperature, and also the time is important. Time depends on the length of your probe, length of your target, and also the type of your the type of probe. Yes, humidity is very important. So this procedure can be done in a liquid environment. So you have to seal your tissue, and you have to cover and seal your tissue in order to keep the concentration of the solution as it is. So the probe incubation can be done by automatic incubators, especially if you're using double-stranded probes. These incubators are very good and give you a standard result. Post-incubation washing is important, because if you don't wash properly your tissue, then you may have some artifacts which could make your life difficult to interpret. And these artifacts can cause misinterpretation or difficulty on interpretation, and these features are much more easy to examine or interpret. For examination, if you're using fluorescent labeled system, then you need to have a dark field and fluorescent microscope. If you're using chromogenic inside hybridization, then you can use your ordinary microscopes. Microscopic filters for fish examination is very important. So they all depend on the probes and the fluoropores that are present on for labeling the probes. So you have to choose the right filter in order to see the hybridization signals. These are all written on the data sheets of these probes. When you buy a probe, you need to, before you're buying a probe, you need to check your filters first. So what is seen, in order to decide whether these the signals are coming from the cellular nucleus, they're not, in order to differentiate them from the artifacts, then you need to know, you need to see the nucleus first. So there are some nuclear dyes, which is most frequently DAP1 or DAP2. You can use them and then when you are examining, you can be sure that the signals are coming from the nucleus. When you're using ordinary microscopes, these filters, you can change the filters and you can see the signals. This is, for example, the target for HER2 news. This is for HER2 signals. This is for reference chromosome 17 signals, and this is the nuclear signals. So when you combine all these signals, you can get these nice pictures, and you can examine and you can interpret your results. These examinations can be done by scanning, sophisticated scanning systems. Even these systems can help you on interpretation or counting your signals. So it depends. You can. It depends which system you have in your laboratory or in your department. You can use these systems for interpretation. But before examining the fish slides, you should remember the original morphology. So if you don't know what the tissue, what was your tissue, then you can get difficulty on interpretation, because this is a dark field. You may miss which cells were tumor cells, which cells were normal cells. So you need to have the memory of your original slide morphology. When you're using cell smears, it is easy to get intact nuclei. So the signals coming from the smears or cytology specimens is much more easy. But when you're working with tissue sections, then you have another difficulty, which on tissue section you may not see all the nuclei of every cell. So you may get some cells, some nuclei were cut, or some nuclei you can lose some nuclei, and also you may have another artifact that when you're looking from this arrow, then you may have superposed false positive signals. So it is important for this. You have to be very careful for interpretation in order to not to count, not to consider the superposed signals. For amplification signals, you need to have a probe for your target gene, and also you need to have a reference, a probe for a reference gene on the same chromosome. So normal signals are usually should be two target reference signal to reference signals, and the amplification is decided the proportion of the proportion between these signals. So there are so many probes, so many type of probes for different amplification detections. So you have to search your probe, or the characteristic of your probe when you first before you interpret or before you examine your slides. If you don't know anything about your probe, then it could be difficult to interpret. So there are so many counting methods. You can have guides for your probes and for the detection systems. So for HER2/neu, there is a guide, and in this guide, it is mentioned that do not count the superposed signals and do not count all these cumulated signals. So this is very important to get the signal of the reference gene and also the signals of the target gene. By FISH, it is also much more easy, maybe, comparing to the fluorescence, but still there are some difficulties on counting the superposed signals. For deletion signals, you need to have a reference gene and also your target gene, probes for the reference and the target gene, but this time you need to have to consider the loss of the signal. So this is a normal signal, two red to green signals in many of the cells, but this is the deleted case, which has got only two reference signals, but only one target signal. This is another deleted case. So the other important issue is the rearrangement, detection of the rearrangement. For detection of the rearrangement, as it was explained before by Dr. Hunt, there are two types of probes. One is the fusion probe, which is designed by two translocated genes. If there is no translocation, then you need to see four different signals, two red to green. And if it is translocated, then the merging of two signals gives a yellow signal. Then you need to have three different colored signals. The pitfalls of these fusion probes, they do not recognize the translocations between alternative partner chromosomes, and also there is always for cell smears there is always a 5 to 10% of false positive fusion signal due to the cellular superposition. But when you go to the tissue sections, this false positivity increases a lot. So for tissue sections, they are not very much recommended. There are some triple signal fusion probes in the market that which are sensitive. If you have the breaking parts of the translocation, if it is a very large sequence, then you need to have, in order to specify it, you need to have several probes. So these probes are designed, for example, for BCR-ABL, you may find some triple fusion probes. Segregation assay or split probes are mostly used for translocation, they are, although they are, they don't give information about which gene is translocated with which gene, but it is, if it is not important, if only the breaking of your target gene is important, this technique is much more preferred for pathologists. So you can show all the alternative translocations independent from the partner genes, and can detect and decrease the false positivity signals due to the superposition. So in normal, the two signals are together, but if it is translocated, then you see separate signals. It is also used for the ALK translocation now frequently for lung cancer cases. And Dr. Diethel's paper also gives some information about how you detect, if you would like to go further to learn more about it. So these were the FISH applications for major chromosomal changes that can be used for our daily routine practice. As I said before, another technique that could be used, the in situ hybridization technique, is the RNA in situ hybridization technique. This is an indirect way of showing that showing the protein expression. But in situ techniques for RNA are not very much easy techniques, as RNA is not a stable molecule, so you can lose your RNA easily. These are not very much practically applied on routine diagnostic pathology, but if you don't have any very good antibody for showing the expression of this protein, then you may use RNA in situ hybridization for showing the expression of the protein. So this in situ hybridization actually is a work of art, and if you have a purpose, then if you would like to know different things, then you can apply different techniques on the same tissue sections. This is an example of a case of PTLD, and in situ hybridization and immunohistochemistry is applied on the same slide in this case. So these HHV8 positive lymphocytes and EBOR positive lymphocytes are around. So if you see these infected cells separately in this case, then you may think that this is an immunosuppressed patient, and she doesn't, or the patient has got an immunosuppressed position and viral infection, but if you see double infected cells, abnormal cells like this, then you may think that this patient has got a tumor, has got a lymphoma, which needs to be treated. So you may use double techniques in order to show your, in order to use your diagnosis as well. This is another double staining, FISH and in situ immunohistochemistry. So you can use these methods together if you would like to show different things. Before ending up my talk, I would like to announce you a meeting which is going to be next year in Istanbul, in the European side of Istanbul. This meeting is the annual meeting of European Association for Hematopathology, and I don't know whether if any of you has participated in one. I would like to invite all you, if you're interested in hematopathology, And also, you may announce this meeting to your colleagues in your departments. This will be a very interesting meeting, a lot of molecular pathology, and will change the diagnostic algorithms of the small B-cell lymphomas. Hopefully will not bring so much confusion in lymphoma classification. Thank you very much, and thank you for your attention.